Let's continue. Antibody versus structure of antibody. These are gamma globulin. These are gamma globulin. Okay. These gamma globulin are mostly produced from B cell. B cell change to plasma cell. B cell change to plasma cells, and plasma cell produce antibody. Okay. So this antibody for defense. So this antibody, our special defense against bacteria, okay, against bacteria. So antibody mediated immunity, our special defense against bacteria. B for bacteria, B for B cells. Keep in mind, okay. For bacterial defense, we have antibodies here. The antibody, when we see the structure of antibody, let me explain in detail. The antibody, the Y shape, okay? Yeah, draw here. So Y shape antibody, and this Y shape antibody having two chains here, okay? This outside chain, so this outside chain is heavy chain, and this inside chain is light chain. So light chain is a heavy chain. Clear? Heavy chain and light chain. So the antibody structure. So this heavy chain have many varieties, there. Okay, this heavy chain. So according to this heavy chain, this outside heavy chain. Okay, this one I changed it to black color. Okay, it's a heavy chain outside. It's a Y shape antibody having Y shape. The two light chain inside. It's a light chain. It's a light chain. The outside we having what? Heavy chain. So the variety. So what are the types there? The types according to heavy chain. Keep in mind, okay? So according to difference in this heavy chain, so immunoglobulin. So they having chain alpha, heavy chain gamma, immunoglobulin IgG, heavy chain gamma, IgD, heavy chain delta, IgE, heavy chain epsilon. Which one left? IgM, immunoglobulin M, heavy chain mu, M for mu, okay, M for mu, A for alpha, G for gamma, D for delta, E for epsilon, okay, E for epsilon, E for epsilon. So we're having 5 different types of heavy chain. Understand? According to variety of heavy chain, we have 5 different types of antibody. It's done. We're having 5 different types of antibody. IgM, IgA, IgG, IgD, IgE. The important antibody here for us, IgM, when any bacteria or you have any type of infection, okay, is just right now started. Innate immunity macrophage, they start working against that bacteria. Then they release some chemical. I already explained the mechanism, okay? They release some chemical like interleukin 8. They call for help to neutrophils. Then they, by their cell, they're moving to CD4 cell and present antigen. How they present antigen, this you guys already know, okay? How they present antigen. By using MSC2 protein represent to CD4. Then CD4 make decision to fight with the help of B cell and produce antibody. The first antibody produced IgM. IgM acute infection. IgM for acute infection. IgG is indicate something chronic. Okay, it indicates something chronic. Keep in mind for chronic infection, for acute infection. IgM for acute infection and IgG for chronic infection. First IgA. IgA is a secretory antibody in GIT mucosa, in respiratory mucosa. The secretory antibody is the IgA. How about the IgD? IgD is not have much important role in our immunity. IgD just act like B cell receptor. IgD just act like B cell receptor. Just present antigen, receive antigen. How about the IgE? IgE for allergy. IgE for allergy condition. Very high level in allergy condition. If the patient have parasite infection, some worms, the defense against the antibody defense against parasite is IgE. So there is a different role of this antibody. IgM for acute infection, IgG for chronic infection, IgE for allergy, IgD for is present on B cell surface. Okay, this just act like B cell receptor. Just act like B cell receptor. And the IgA, what's the importance of IgA? IgA is also known as, keep in mind the name of IgA. IgA secretory antibody. IgA secretory. So this antibody, IgA not present in blood. IgA not present in blood freely, okay? So where's IgA? IgA, GIT mucosa, oral cavity mucosa, reproductive tract mucosa, respiratory tract mucosa. So this IgA on mucosal surface. IgA on mucosal surface. Clear or not? IgA on mucosal surface. We just assume IgA enter to blood. If IgA enter to blood, it's not good for other body cells. It's not good for other body cells. This IgA, if enter to blood, normally I am just explaining here. IgA is just a secretory antibody. It's secreted along with mucus, only on mucosal surface. Okay. Reproductive tract mucosa defense IgA. Respiratory tract mucosa defense IgA. GI tract mucosa defense IgA. So IgA not present in blood. If IgA enter to blood, damage vessel. IgA causing vasculitis, inox swollen purpura. Let me explain regarding IgA. Okay, IgA damage kidney, IgA nephropathy. So IgA never enter to blood. Keep in mind. If IgA enter to blood, this IgA is engulfed by macrophages and clear from the blood. IgA is not allowed to freely move in blood. Understand? So how about the IgM and IgG? This part infection. If body have IgM, it means the infection in acute phase. Okay. If the IgG there, it indicates some chronicity. Understand? So how about the IgE? IgE, IgE for allergy, IgE for parasite. I just give you overview of the antibody, okay, how they're working and what are the function of them, the general function there, okay. IgM, IgG, IgE, IgD, which act like a B cell receptor. IgM also act like B cell receptor, okay. So we also have, but IgD not have any other function, known function there, only act like B cell receptor, okay. So very important function about IgA is also known as security antibody. The mother, when the mother starts breastfeeding, feeding her child, so that milk, breast milk also contain IgA. Okay, so just for defense. Understand? So it's the IgA, it's the IgD, IgG, IgM. Let's continue. In this chapter, first we see about their structure, okay, and what are different type of region there, and how they're working, what are their type, isotype, allotype, idiotypes, and what are the function of antibody, okay, one by one. Antibody, they produce from B cell, clear? The B cell, stimulation with antigen and lead to differentiation of B cell. B cell change to pre-B cell, pro-B cell, pro-B cell, pre-B cell, again pre-B cell, again immature B cell, again mature B cell change to plasma cell, yeah? So plasma cell, Plasma cell producing what? Plasma cell produce antibody. Understand? So what are the antibodies? These are the some glycoproteins. Okay, these are the proteins. What type of protein? These are the gamma protein. Keep in mind. What protein? Gamma protein. All are the gamma protein. Immunoglobulin Ig, immunoglobulin IgA. Clear? Immunoglobulin IgD, like this. Okay. So these are the all are gamma protein. So antibody doing what? Antibody bind with the specific antigen. The specific antibody have lot of variety. We have thousand million of bacteria there. Our antibody recognize them separately. Okay, much diversity in this antibody. Antibody is secreted into serum. Okay, secreted into antibody mostly secreted into serum. Which antibody is not in blood? IgA is not in blood. Keep in mind. It's not present in serum. I'll explain. Okay. All other antibodies they present in blood, but this IgA not in blood. Understand? Okay. Antibody also called immunoglobulin. Understand why they called immunoglobulin? Immunoglobulin. These are the defense protein. These are the gamma globulin. Understand? So immunoglobulin. These are the globulin protein. Beta globulin, alpha globulin, gamma globulin. These are all protein are gamma globulin. Globulin protein working for immune system is a globulin protein. Yeah, globulin protein working for immune system. That's why immuno working for immune system. Immunoglobulin is a common name for antibody. Ig, immunoglobulin, immunoglobulin. Which globulin? Alpha, beta, gamma. It's a gamma globulin. A secretory Ig, immunoglobulin, membrane Ig, B cell receptor. Secretory, some are secretory. Which one is secretory? IgA. Okay, keep in mind, IgA. And some antibody are they act like B cell receptor. Immunoglobulin B cell receptor. Which one? IgD. IgD here, and this is your secretory is your IgA. Remaining we having three more. IgM, IgG, IgE. So these three antibody always circulate in serum, always present in serum. Now you understand? Three antibody IgM, IgG, IgE. We need these three for defense for allergy. Okay, IgE, IgM, IgG. So IgA is on mucosal surface secretory antibody, and IgD is just act like B cell receptor. Understand? Clear or not? Their location clear? Antibody in blood, IgE, IgM, IgG. Okay. Antibody present on B cell surface, IgD. Antibody is not present in blood, just secretory antibody, secrete along with mucus. Okay, on mucosal surfaces, just on mucosal surfaces, GIT mucosa, respiratory mucosa, reproductive tract mucosa. Okay, so there we having what? IgA. Done. Their location clear? Okay, let's see. The blood contain three globulin there. This well done. Alpha, beta, gamma. Yeah. So what are the antibody? These are the gamma globulin. These are the gamma globulin. Alpha globulin, liver produce albumin. Liver produce alpha beta globulin. In our body, we also have gamma globulin. This gamma globulin come from B cell, and this gamma globulin come from B cell. It's very high. It's higher than alpha and beta. Okay, it's a globulin protein. Okay, let's see. Let's talk about what's the structure of antibody. The structure of antibody. We're having four chains there. How many chains? Four chain. Two heavy H2. Two heavy chain and two light chain. Okay. Two heavy chain and two light chain. And what's the connection between them? The connection between them, disulfide bond. Okay. The connection between them, like it's a light chain, it's a heavy chain outside, heavy chain. Here the connection. Okay. This connection is disulfide bond. It's a light chain. It's a light chain. Connection with disulfide bond. Clear? So that they are light chain, heavy chain. They connected with each other with the help of disulfide bond. It's a disulfide bond. Which amino acid have sulfur? The biochemistry. Which amino acid have sulfur? Important here for disulfide bond. Cysteine amino acid. Which one? Cysteine amino acid. Okay, the cysteine amino acid. The polypeptide chain 25,000 molecular weight. Okay, two identical heavy chain you can see 50,000 molecular weight. 70,000 is a heavy chain, it's a light chain. Understand? So heavy chain having five isotypes. Heavy chain. I already explained our antibody classification depend upon heavy chain. Okay, so heavy chain alpha, heavy chain gamma, heavy chain mu, heavy chain delta, heavy chain epsilon. Epsilon for E, delta for D, mu for M, gamma for G, A for alpha. Yeah. So these. So for our classification, the different types of antibody, the variety is that the variety depend upon the heavy chain. We're having heavy chain mu. If the heavy chain is mu, then IgM. Production of is IgM. Heavy chain gamma is the IgG. Heavy chain alpha is the IgA. Heavy chain epsilon is the IgE. Heavy chain delta is the IgD. Okay. Okay. Let's see the light chain. Light chain, the two types. Light chain having two types. One is kappa. One is lambda. One is lambda. Lambda, and one is kappa. There are two types of light chain, five types of heavy chain. Understand? Five types of heavy chain, two types of light chain. Now see the heavy chain and light chain. Okay. So green in color here, you can see it's a heavy chain structure. Okay, it's a heavy chain structure. Clear? And yellow in color, it's a light chain structure. It's a light chain structure. Heavy chain have how many parts? It's a heavy chain 1, heavy chain part 2, heavy chain part 3, heavy chain part 4. Oh, we're having 4 parts there, okay? Keep in mind, this part of antibody known as FC part, okay? This part of antibody known as FC part, and this part of antibody known as FAB part. FAB. Antibody according structure, okay? The light chain done. Heavy chain, light chain, heavy chain, light chain, okay? Let's see according to function. Now we divide antibody to part. One is, what means C? C stand for constant. C stand for constant. What's the meaning of F? F stand for fragment. Fragment mean piece, yeah? Constant fragment. Constant fragment. Constant mean fix, yeah? Constant fragment is FC. Constant fragment is FC. What's the FAB? Again, F mean fragment. F mean fragment. And what's mean AB? AB stand for variable. Variable, okay? AB is AB word for variable. This AB word used for variable. Understand? Clear? This part is variable fragment, and this part is constant fragment. Done or not? This part is variable fragment, upper part, and this Y arm, this arm, and this arm, like this antibody, like this, okay? Y shape. So this part is constant part, and these two arms are, these are two arms, yeah? So these two arms are variable part of antibody. Okay, let's see how the constant part is formed. Which chain form the FC segment? Heavy or light chain? Which chain form the constant segment? Heavy or light? Just heavy. Okay, so heavy chain form constant part. What is the function of constant part? Constant part for binding with cells. Keep in mind. Constant part for binding with cell. Okay, binding with cell. This part for binding with cell, clear or not? The constant part, for example, is a mast cell. It's the mast cell. The mast cell. How antibody attach with the mast cell? This part, antibody attach like this. Which part attached to mast cell? Tell me. Which part attached to mast cell? With the cell. Which part? FC, okay? So this lower part is this straight part. FC segment, FC segment, FC segment. Understand? FC segment attachment with the cell membrane, okay? Attachment with the cell membrane. Another thing, this FC segment can stimulate signal inside, okay? This FC segment, second function of this FC segment. FC segment help to activate complement system. Help FC segment help to fix or activate, okay? Help to fix, what's the complement system? Later, one lecture on complement system later, okay? Help to fix complement system. It's a very important system for defense, complement system, okay? Complement system protein, 20 protein system, okay? FC segment for complement system, FC segment for attachment with cell. Listen very careful, okay? FC segment, if antibody attach with FC segment, is just a binding. Can antibody kill that cell with FC segment? No, 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 no. Antibody, they're not killing if they bound with, like if they attach like FC segment attached with the membrane, it's not a killing way. Okay, listen carefully, okay? Another cell, if antibody attach like this, can you tell me which part attached with the cell now? This is the cell. Now which part attached?
FAB.
FAB, okay? So now this part is attached with the cell. Oh, FAB part. So this part is a variable part, okay? So this part, you see here binding site? It's a binding site. 4 binding sites here. 4 binding sites. These binding site for antigen. Now antibody, if attached with the cell, antibody FAB for antigen. This antibody consider this full cell as an antigen. If antibody attach like this, it's mean antibody going to kill that cell. Understand? When antibody in fighting mood, how antibody attach? FAB attached with that area, okay? FAB attached with the bacteria, FAB attached with the cell, killing. It's in a fighting mood, yeah? If not, just let's continue, okay? This difference clear between the attachment of FAB and FC. When antibody in fighting mood, how they attach? They attach like the arms attached with that area, okay, with the bacteria. How antibody attach with the bacteria? If the bacteria, antibody attach with bacteria, how attach with bacteria? Antibody attach with the bacteria with the help of FAB segment. Understand? With the help of FAB segment. Clear or not? So okay, let's see now again, come back. There's a difference between two parts. Clear? Difference between two parts, FAB and FC. The two part of antibody, one part is a constant fragment, one part is a variable fragment. If antibody using constant fragment for attachment, it's not a killing, it's just maybe stimulation or activation, okay? If the antibody using FAB part for attachment, it's antibody in fighting mode, okay? Antibody in killing mode. Clear? Antibody in killing mode. It's a variable part. Okay, let's see what a different area here. So this is the constant part. For the constant, I am using word C. For the variable, because this part is a variable, 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 variable. Understand? These two part here is a constant, constant, constant. Let's see the structure now more clearly, okay? Let's see the structure here more clearly. How we classify this, okay? The constant, how many part are constant here? How many part are variable there, okay? this part, this full fragment is, listen carefully, this full fragment is FAB, full part is FAB, and this full part is FC, is a constant part. Understand? This part is a still constant part, not take part here, okay? Not take part in fighting. So okay, not only this part is important for us. This part is important for us, okay? Okay, what are the things here? You see, constant H1, this segment, constant H1, move down, constant H2, constant H3. What's mean H? H for heavy. H for heavy, okay? This part is a constant part of heavy chain. Constant part of heavy chain. Constant part of heavy chain. How many parts are constant for heavy chain? 3 parts are constant. CH1, CH2, CH3. Okay, let's see. The last part of heavy chain. This is the full, what is this chain? Heavy chain. This chain is full in heavy chain. Clear or not? Okay. This last part of heavy chain is the variable part. So that's why I'm using word here VH. Variable part of heavy chain. Constant part of heavy chain. Constant part 1. Constant part heavy chain 2. Constant part heavy chain 3. Understand? Labeling done or not? Let's see here. It's a green is heavy chain, okay? This is a green one. Heavy chain. It's a green one. Heavy chain. The green one. Heavy chain. It's a 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 heavy chain. Done? How many constant parts? This one is the constant part here. The constant part. Clear? It's the constant part. It's the constant part. It's the constant. How many constant parts? 1, 2, 3. Start counting from here. CH1, CH2, CH3. Which part of SFC segment attached with the cell membrane CH3? Understand? CH3. CH3. Okay, let's see now talk about the light chain. Okay, this is the light chain. This one is light chain. Light chain having two parts. One this one, another this one. Okay. So this light chain having one constant part Cl and one variable part VL. VL variable light. VH variable heavy. Understand? VL variable light chain. VH variable heavy chain. Clear? Cl. What's a Cl? Constant light chain part. Constant part of light chain. Constant part of heavy chain. We just have constant part of light chain is a one constant part. Constant part of heavy chain, because heavy chain is very bigger, yeah? So heavy chain is very bigger. One part, then another part. Okay. So one heavy chain part, second heavy chain part, third heavy chain part. So this third heavy chain part, here we having inside we having what? Inside we having light chain here. It's a light chain here. Here is the light chain here. Clear? For the constant part, it's a constant part heavy chain 1, constant part heavy chain 2, constant part heavy chain 3. Okay? Clear? This is a constant part of light chain. Constant part of light chain. Let's see the variable part now. What's the variable part? This one outside is a heavy chain. The inside, this one is a light chain. Outside is a heavy chain. The inside here is a light chain. Clear or not? Okay, I change the color here for the light chain. Light chain I change to green. Okay, with the full light chain. This is a full light chain. Okay, the inside full light chain. So light chain having two parts. One part is a constant light chain. Another part is a variable light chain CLVL. Okay. How about this part? This part is a variable heavy chain. Understand? Partition done or not? Everyone clear about the partition? Different parts clear or not?
Yes,
Constant,
sir.
Yes, sir.
constant fragment, variable fragment. Then we further explain this fragment, okay? So these fragment, they having the constant and variable part. So just a labeling here, okay? The CH1, CH2, CH3. Start one from here, okay? CH1, CH2, CH3. However, last part is important for antigen bonding. This part for antigen bonding, understand? How about this part? This part for binding with the cell membrane, just for stimulation, not for fighting. Now you can understand which part for antigen binding. Variable light chain, variable heavy chain. This part for working for antigen. Antibody part, variable part of antibody attached with antigen, understand? Variable part of antibody attached with antigen. Which part of antibody attached with antigen? Variable part. Light chain variable part, heavy chain variable part. Why I mention here variable, variable, variable? I am using constantly this word variable, variable, variable. This part have lot of variety, lot of variety. One bacteria enter your body, the variable part, that protein part